everyone that is in this conference. I will present first the Dr. Marco Ferrante. Marco Ferrante is a postdoc of a project here in the Azorean Biodiversity Group from C3C, working on ecosystem services. It's working within the project agroecosystems that is funded by Azorean government. Marco has already some experience in experimental ecosystem services using sentinel approach. You will talk today on specific experiments conducted during the last year in the Azores, and we expect to see some interesting novel approaches in this field of ecosystem services measurement. As you know, usually ecosystem services in some cases are measured based on indirect approaches, but as you will see here, we will have a very quantitative way to measure directly ecosystem services and these services in real habitats, in real agroecosystems. I will give now the place to Marco to start his presentation.
Thank you, Paolo. Can you hear me? I'm going to share the screen. Let me see. Can you see my screen?
Yes, yes.
Okay. So thank you very much for inviting me today. As Paolo mentioned in this presentation, we are going to talk about the methods to assess ecosystem function. We are going to start defining what ecosystem functions are and their relationship with biodiversity that provides them. And then I'm going to show the sentinel approach as a way to measure ecosystem function and also its application in the agroecoservice project here on Terceira Island. So first of all, let's clarify the terminology. In 1935, the word ecosystem was coined by Arthur Puzzley, the plant ecologist. He wanted to refer to the biological community, to the biocenosis, together with the physical environment, the biotope. Ecosystem function are all the physical, chemical, and biological processes that translocate and also transform energy and material in ecosystem. Essentially, they are the outcome of the individual activity of species. Think, for instance, about decomposition or photosynthesis, but also the interaction between species, such as predation, herbivory, parasitism. And because some of these processes are extremely important for human welfare, such as water availability or the regulation of the atmosphere composition, sometimes the word ecosystem function is used as a substitute for ecosystem services, which are all the benefits we get from ecosystems. However, it's important to distinguish between them because ecosystem functions are not end products. They are just intermediates to the production of ecosystem services. Let me do an example with this jumping spiders here. Imagine that we have a community of jumping spiders. This is our biodiversity. And spiders are predators, so they will provide the function of predation in ecosystem. In agricultural environment, predation on pests is an ecosystem services, it's pest control. At the same time, so you can also have predation on another predator, another natural enemy, which is intragil predation, and this can be considered as an ecosystem disservices. So you see that in the ecosystem services concept, there is a perspective which is completely utilitarian, and this is not present in the ecosystem function concept. The relationship between biodiversity and ecosystem function is one of the fundamental questions of ecology, and essentially dates back to Darwin, was already talking about the fact of having more than a single grass species in English gardens. We know that if in an ecosystem we don't have species, let's say that imagine that biodiversity equals 0. Of course, we won't have any ecosystem function. Now the question is, what starts to happen once we add species to the system? Let's look at this graph here on the right. We see that we have ecosystem function on the vertical axis and biodiversity on the horizontal axis. If species are mostly unique, they perform unique function, unique services. Adding them to the system will make the system function biodiversity relation look linear because there will not be any overlap between them. However, you can also imagine that species are redundant, that they perform similar functions. And in that case, you should reach relatively quickly a plateau, and then the service or the function will stabilize. You can also have a situation in between, and in reality, there have been many models suggested to describe this relationship. And here I'm only showing in this graph on the left some of the most famous. You may think that this is only a interesting theoretical question, but nowadays this is extremely important because of the current biodiversity decline, because you may also read this graph backward. What starts to happen to ecosystem once we start removing species? Do we make them collapse? Will ecosystem functions stop working somehow? In the beginning of the 90s, there have been a few studies started to trying to understand this relationship. First of all, there was a group working in the Imperial College of London, and then colleagues in Minnesota at Cedar Creek. And this study confirmed a positive effect of biodiversity on ecosystem function. However, there were a lot of limitations. These were control experiments. They were done on a very small temporal scale, on a very small spatial scale. Most of the study came from North America or Europe, working in grassland, measuring only ecosystem function provided by plants. Essentially, we don't know how to translate these results into real ecosystem out there. In agroecology, or in general in agroecosystem, we are particularly interested in identifying the species that provide ecosystem function and also to support them, to enhance their habitat. Most of the species are arthropods. Imagine like pollinators, natural enemies, and traditionally in entomology, we collect arthropods, we kill them, and then we identify and describe a community using biodiversity indices. Let's make a mental experiment. Let's imagine that we have a cereal field and we release natural enemies, and we double the density compared to the original one. Does it mean that we're also doubling pest control? And the answer is not necessarily. I'm going to show you a practical example from a recent study. This was done considering two predatory guild communities. In black, we have spiders, and in gray, ground beetle carabates. And on the vertical axis, we have their density, the relative abundance, let's say, and the horizontal one, we have the body size. And then on the right, there is a third panel, which is the predation rate on aphids, pest control. And in the top graph, you see that we have high density of spiders and relatively low density of ground beetles, and predation rate on aphids is high. In the graph below, we have the same density of spiders, but then the density of large ground beetle increases and predation drops almost by half, which sounds counterintuitive. We increase the number of big carabates and predation decreases. And that's most likely because ground beetles are opportunistic predators. So they will also predate on spider, and therefore the predation by spider on the aphids will be reduced. So this is just to tell you that it's not so straightforward that we have a more diverse community of predators, or more abundant predators, and automatically we get more of the function, more of the service. And this is why in recent years, we have been trying to bypass biodiversity measures and try to look at this function directly. There's been an important review published in 2015, and 2017, Professor Lovey and I published also a review on the methods to quantify arthropods predation in the field. No, the advantage of this direct measures compared to the traditional biodiversity proxies are multiple. So first of all, as I mentioned, they are direct. So we don't have to look at biodiversity because it's difficult to interpret what a change in density means for the service. Second, if you think from the farmer perspective, the farmer is not really interested in increasing the number of pollinators or predators. He is interested in the service. There is also a third aspect that is important, which is the taxonomical knowledge. When you work with biodiversity, you have to identify species, and there are not always taxonomies to do that. In this case, when we work on ecosystem function, we don't need taxonomical knowledge. Also, these methods are essentially all non-lethal, so we don't have to kill what are beneficial insects. And they're relatively simple and easy to use, which makes them suitable for citizen science and global experiments. I'm gonna show now the application of this sentinel method. You will see a bunch of them in the agri-service project on Terceira Island in the Azores. We are located in the middle of the Atlantic, and this is a big project. We basically selected 6 habitats that are characteristic of the island. We work in the native forest, we work in vineyards and orchards, low and high elevation maize fields, and also pastures. And we selected 3 sites for each of these habitats. And we measure different functions. We measure herbivory rates on a crop plant that can be considered a disservice. We measure predation rate on a sentinel prey that emulates a pest, that emulates a caterpillar, which means essentially pest control is a service. We also measure pollination of crop plants, which is another ecosystem service. We measure seed predation on wheat and a weed seed, which can be considered respectively an ecosystem disservices and the services. And we also measure the composition of organic material, which is a service. In this presentation, I'm only going to present preliminary results on in vineyards and orchards. If you've been on Terceira Island, you know that vineyards here are very special. They are basically growing in a very small enclosure, sometimes even 4 or 5 by 5 meters. And the plant grows very low. The farmers create some hole, you see, where to root the plant, and they raise wall of volcanic rock to protect the plant from the sea wind, especially from the salt that will kill the plant. And it's a particular habitat also because it's not really stable. For most of the year, the biomass, the plant biomass is very low. If you walk in vineyards, you normally see rocks, and especially at the beginning of the season, like in spring, you don't have almost any cover. Only during the harvest later on in September, in autumn in general, or at the end of the summer, there is much biomass. In contrast, orchards are very complex habitats. They are 3-dimensional. They are characterized by dense undergrowth, which creates favorable microclimatic conditions for ground-active insects. And here on Terceira, they're also mostly mixed crop. Often they are in combination with banana, sometimes other minor crops like apples, guayaba. So these are like a mosaic, it's a polyculture. Now I'm going to show you the methodology we use for each of these ecosystem function in general, or ecosystem service and disservices. We use the sentinel approach, which means that we expose a sentinel, can be a plant, can be an insect, can be something that emulates a component of the ecosystem. And the sentinel approach means that we are emulating a process. We emulate herbivory, we emulate predation, and we do it in a way that is standardized so that we can get comparative measures. So the idea is not to get the real absolute predation rate in vineyards or orchards. The idea is to get comparable data in a standardized manner. Okay. Now we start with the herbivory. So for herbivory, we have grown in the greenhouse lettuce, and about a month and a half later, we exposed them in the field. We dig in the vineyards and the orchards, and we left them there for a couple of weeks. And after a couple of weeks, we collect them, and I visually estimate herbivory rate on the leaves of the lettuce. I also tried to use a program, taking photo of the leaves and estimate it more easily with an application. Unfortunately, we had some problem because when you take a photo and the leaf is not flat, there will be shades, and many programs interpreted this shade as a herbivory, like a missing part, because it's a different hue of the green. So we've done that visually. For predation rates, we use a method that I used in the past. I've done my PhD in Denmark, working mainly on this, and basically fine-tuning the application of this method in different ecosystems. We basically create sentinel prey using non-toxic plasticine, and this emulates a real caterpillar, a real green caterpillar. It's a normal coloration, very common in almost every environment, and indicates palatability. Normally, green caterpillar are not toxic. Let me see if I can try to show you a video of how to produce this sentinel prey. Okay, I think it works. So you see that with the non-toxic plasticine, and this one here is a garlic press. It's a modified garlic press. I have cut the bottom of it, and then I place one Danish crown inside. The current in Denmark has a hole, and this allows you to basically put a plate of plasticine inside and then squeeze it. And once you squeeze it, you get a long green snake, and then you can cut the caterpillar of the size you want. There is a main difference, a major difference with the real prey, because sometimes people also use real caterpillars. They pin them or they glue them. The fact is that when you use real prey, the predators will consume them, will attack them and take them away, and then you have no idea who was the predator. With this technique, you get some information about predator identity, because the predator will attack the caterpillar, will left a bite, but then will not take it away because it's not food. So you see that this is the green caterpillar snake, and now we're cutting this. We have to roll it to be sure that there are no previous marks on the plasticine. And then here you have your caterpillar. You can do several things with it. In this experiment, we glue it to a piece of reed or bamboo. You see it there. We use just a drop of super glue, and that's because we wanted to expose it at ground level. So once we glue it on the piece of reed, we don't have to touch the plasticine directly. It's also easy to handle it. In my past experience, I've done different things with it. You can also put them directly on leaves. For instance, here Is everything okay? Okay, I think it disconnected for a second.
I lost internet in the university for a moment, but it's okay.
Okay, we're back. I hope we are still on YouTube. I can't check that. Okay, let's see for pollination. Pollination is an important ecosystem services, and it is very often measured with density and diversity of pollinators, because it's not an easy service to measure directly. We try to do so. We use phytometers. We expose plants that need insect pollinators. In this case, we use strawberries. So we've grown strawberries in the greenhouse, and then we expose them in two setups. We have this cage, you see it with this large mesh. This allows bee and diptera and other pollinators to get in easily. And then we have another one, sort of control, where we use a tight mesh and we stop pollinators to come in, so that we can compare between the two. Unfortunately, in the orchards, we had some problems setting up this control. So in the results, I'm only going to compare the seed set between the accessible cage in the two habitats. When I see the seed set, I say the seed set is because what we are going to do is to expose this plant for a couple of weeks to pollinators, and then we brought it back to the glass house, and we left it maturate, and we get the fruit. And after that, I photograph the fruit, and I start counting with a program all the mature seeds in it, all the achenes of the strawberry. So you get a direct measure of the benefit of pollination, because after all, for the plant point of view, that's the number of seeds. For seed predation, we use another low-tech technique. We modify Tupperware. We cut two openings on Tupperware. You see that we have two set up also here. We have one that is open to both vertebrate seed predator, rodents mostly, and invertebrate seed predators, insects. And on the right, we exclude the rodents with a plastic mesh, so that we can only discern what's the contribution by invertebrate seed predators. And on top, okay, we also use two types of seeds. I mentioned that for this service we measure both service and this service. We have wheat seeds on the left and dandelion seed taraxacum on the right. Taraxacum is a weed in most crops in most continents. And we use double-faced sticky tape, and we place 5 rows with 10 seeds each. And then we sprinkle some soil so that the predators can enter the trap without getting stuck on the tape, and they can feed on the seeds. And here you see it open, but of course we use a cover to avoid rain and to avoid birds. And the trap was exposed for 2 days, and after 2 days it was collected, and I can calculate the percentage of seeds that are missing or that are damaged by predators. Finally, we measure also decomposition, and we use a relatively recent technique, is the Teabags Index. We measure the weight of Teabags with a precision balance in the lab, and then we expose them in the field, dug into the soil 10 centimeters deep, and we left them there for 90 days. And after 90 days, we collect them, we dry them and use with the oven for 2 days, and then we weigh again the organic material, and we can see how much mass was lost during this day for microbial activity. I wanted to show you some preliminary results. You see here there is also some spoiler for the other habitats. Just look, orchards and vineyards. You see that in the orchard, herbivore rate was significantly higher than in the vineyards. It was 7 times higher. I will mention later why we think that happened. For predation or pest control, we found out that the main predators were vertebrates, in particular birds. Almost 75% of the predatory marks, the attacks were due to birds attacks. And you can identify them clearly by the mark left on the caterpillar. You see, this is a VU mark, which is the pick, the bill of the caterpillar. Sorry, the bill of the bird on the caterpillar. Here on the right, you see a mammal mark. Rodents accounted only for about 8% of the attacks. These are also very easy to identify. You see the teeth. These are the incisives. And vertebrate predation was significantly higher in vineyards than in orchards. Invertebrates were relatively low. They surprisingly low. In my experience, they are normally higher, but here, perhaps because it's an oceanic island, the diversity is lower and many large ground beetles are missing. We only found that 15% of the caterpillars were attacked by arthropods. And predation rate was relatively low in both habitats, and there were no significant differences. Another spoiler alert for the seed set, just to look at the orchards and vineyards. Interestingly, we found that the seed set in vineyards and pollination service was significantly higher than in orchards. Seed predation is a little bit tricky because we found out that most of the seed box didn't have any signs of predation. Almost 80%, around 80% of the seed box were intact, no predators enter. But sometimes if a predator enters the seed box, then they consume a lot of seeds. So seed predation is very patchy. We haven't seen any significant difference between the two habitats, and not even between the two seed species. But there was a difference between predation in open box that are accessible to every predator versus the exclusion one. Finally, about the decomposition, we haven't seen a difference between habitats once again, but the mass loss of teabags was higher than the mass loss of the rooibos bag that we also use in pairs. So we haven't seen a difference between habitats, but the quality of the organic material matters. This is to summarize the results. So essentially, we found the significance only for herbivory, vertebrate predation, and pollination. The two services, vertebrate predation and pollination, were higher in the vineyard, and the ecosystem disservice, herbivory on the lettuce, was instead higher on the orchard. We think that habitat complexity may explain herbivory because of the dense undergrowth that I show you in the original picture. There were a lot of slugs in orchards, so they found possibly favorable microclimatic conditions that are lacking in vineyards. In vineyards, we almost didn't see any slug and snails because the soil is barren and is basically very dry, like a desert. For what concerns vertebrate predation, which was mostly a reminder by birds, we think that possibly this can be explained because of the background, because grain caterpillar on the black volcanic rocks are relatively easy to spot, and the birds are there, so they find them more easily and they try them, they attack them. So the mortality rate of a caterpillar will probably be higher in vineyards because of this. For pollination, that was in a sense a surprise because I was imagining that being more complex, the orchard would attract more beneficial insects. In fact, what may have happened is the opposite. That's because there is almost nothing in vineyards. There are no resources. All biodiversity concentrates on the few resources that are there. So when we exposed the strawberries there, the pollinators didn't find anything else, and they all went there. Instead, in the orchards, pollination may have been diluted also with the orchard tree and the other crops. I finish with this, and I want to thank you again for inviting me here, and I'll leave you with this table of summary results, and if you have any questions, please let me know. Thank you again.
Thank you, thank you, Marco, for giving this nice presentation, highlighting also the difficulties of having all these kind of experiments done with preparing all the plants for herbivory, for pollination, and setting up these quite it seems simple, but in practice they are complex experiments to have everything standardized. And okay, thank you. And let's see if we have questions from the participants. Marianne, you see if there is any question in the chat. For now, there is no questions. Okay, I want also to highlight also a very important thing is that now people, researchers in this area are trying to propose protocols, standardized protocols for this kind of experiments. The Teabag experiments are already very well known and with a long history of standardization, but still there are some challenges due to the fact that the producers of these tea bags are always changing the structure of the tea bags and these kind of things. But for the other experiments, there is also the need to have standard protocols that people could apply, and in future meta-analysis could compare experiments. The sentinel predatory experiment already has some good experience with the people comparing latitudinal trends in predation rate and these kind of things, but there is a need to do for all these kind of services standard protocols. I don't know if Marco want to add something to this.
Yeah, absolutely. I think what you say is very important. In fact, if you think that comparing biodiversity between biomes and ecoregion is really difficult because it's of course unique to the different places, but comparing ecosystem function with the same methodology is much easier, and that's why in the last 3-4 years they published an increasingly number of papers that try to assess some of this function globally. And one of these was published in Science in 2017. The authors used the sentinel spray on a global scale at different latitudes and different habitats. And they found and confirm what is another one of actually of the few, I dare to say, laws of the ecology, which is the latitudinal gradient, that biotic interaction increase to the equator and decrease going towards the pole. These are things that have been around for decades. The theory, we also have some biodiversity data that suggests that, but somehow a standardized confirmation of this only arrived now, thanks to this kind of technique.
Marco, there is a question from Hernan. He says that he's very nice, his experience, and ask, did you use spectrophotometry to compare sentinel larvae with real larvae? I mean, for the real vision of the predator.
No, in this experiment we haven't done it because we were not trying to assess predator behavior or preference to different color prey, but there are studies comparing different prey phenotype, usually coloration, and these are done by groups that work with they try to work with aposematic coloration and try to explain why certain predators prefer certain types of prey, and they do this kind normally this kind of test to have an idea of what predators actually see, because they clearly see differently from us. But for our purpose, the green coloration was just because this is a common coloration in nature. It doesn't indicate any toxicity, so it should be, as I say, should not scare any predator, and they will be willing to try it. So it's also an easy one for comparison, for standardization.
Another question now from Pedro Pinho. Great approach to measure directly ecosystem service as possible. One question: did you manage to standardize the surrounding habitats? For instance, the same type of surrounding ecosystem in vines and orchards?
Well, we have tried to select sites that were the most similar as possible to each other, but the two habitats were different, and actually we want them to be different because our idea was to compare a complex habitat versus a simple one. There is this idea in agroecology that complexity is beneficial to biodiversity. I haven't mentioned it, but the agroecoservice project is fairly big, and we also collect measures of biodiversity. We're also trapping insects and identifying them. So I take it advantage of these notes to underline that I'm not claiming that these methods are substitutes for biodiversity methods. They can be used in a complementary way. We wanted to compare a complex habitat versus a simple one. So when we look at the orchards, we look orchards that were similar to each other, but we want them to be different from the vineyard.
But the question is if, but the question mark, if is the habitat, the matrix of habitat surrounding vineyards.
Oh, surrounding the crop. Surrounding the
crops are the same. Oh, and indeed,
okay. No,
indeed, they are not the same because vineyards are in a specific microhabitat situation in the Azores, and orchards are more embedded in urban, urban environment. But in both cases, is low elevation, is near sea level habitat. Yes.
They are two habitats that are both at low elevation and experience similar temperature and precipitation regimes. Also the urbanization thing, you know, we are in the Azores. They are not really big cities, so yes, orchards are somehow embedded or closer to houses, but it's not that urbanization is a major driver here of disturbance. And also, actually, the vineyards are always associated to houses, and sometimes are summer houses, but the vineyards are all concentrated in one area in the north of the island, while orchards may be found a little bit everywhere.
No more questions. Okay. Okay. We will wait a little 1 minute only to see if there is someone who wants to make a question. I want also to remember that Pedro Pinho asked, does you use proxies of ecosystem service instead?
Sorry, can you repeat, Paulo?
Does you use proxies of ecosystem services in alternative?
Other alternatives to the sentinels we use?
Yes.
Well, yes, absolutely. For sentinel prey, you can use other things. You can use real prey. You don't have to use plasticine prey. Plasticine prey are somehow have some advantage because, as I mentioned, they allow you to identify the predators, and we were interested in this aspect. But if you want a setup that somehow is more realistic, you should use real prey. Sometimes people use real caterpillars or aphids. In aphid cards, they glue aphids. Sometimes they even glue eggs on the card, and then they do the same. They expose them for a set time, and then they retrieve them and calculate how many eggs were damaged. For the pollination, we wanted to use strawberries because it's a plant that is also insect pollinator, and it's also a plant that is grown here, so somehow is as a service, is something that can be found in this kind of a habitat. But sometimes people also use ornamental plants. They are usually easier to buy, and you can grow them possibly easily. And some of these ornamental flowers have a lot of ornamental plants have a lot of flowers, so you get a lot of seeds and you get a lot of data to analyze. For herbivory, normally herbivory is assessed directly in the field, but not in a standardized way. I don't know many other studies that expose a plant like they expose a plant for pollination, and then they retrieve it from the field. They normally go there and collect a number of leaves and assess it directly into the field, which has the problem that you don't know when the herbivory happened, because the plant was there all the time, and it's difficult to compare with herbivory rates in other habitats. For decomposition, yes, instead of tea bags, sometimes people use leaves or other organic materials. They put them in bag. The concept is the same, and they dig it into the soil and wait for months to see how long it takes to decompose. So yes, there are other alternatives. We have tried to use the ones that are easier to standardize.
Okay, we have another question now from Ricardo Rocha. He says, amazing stuff. Congratulations to Marco and the team. I wonder if you have any thoughts on how this pattern found in the Azores, in insular setting, might compare with the same ecosystem functions in mainland. Indeed, Marco has already made this in mainland. Can you say something about that?
Okay, so first of all, for the habitat themselves, I think it tells relatively little, because orchards and vineyards are both very special here on the Azores. You don't find vineyards growing this way in mainland, and also orchards are normally monoculture. Here is very special in the sense that they are grown in a very unique way. However, I think that the results reflect in terms of complex agroecosystem versus simple agroecosystem. I think that you have several ways in mainland to increase complexity. Ecosystem, all conservation biological control is based on that, essentially habitat manipulation to favor beneficial arthropods mainly. And the idea is to create a complex habitat like orchards here, instead of having a simple habitat like vineyards that are relatively simplified. Of course, the scale is also different, and this is an oceanic island. There are sometimes functional groups that are missing. We have a lot of invasive species, a lot of problem with pests. So it's a setting that, in a sense, is unique, but possibly, probably in mainland you get more species. It will be also more difficult to identify who are the responsible for these services. Here we have a very Paulo in the last 30 years create an incredible database and knowledge of the arthropod fauna of the Azores, which is unusual, I have to say. Normally, other groups don't have this background information. So we have the advantages. We can guess, looking at the traits of species, also which one are the ecosystem service providers. And this is not easy to translate in a mainland situation.
Mario says, very nice work with use effort. Can you address the limitations of the sampling design approach? For example, predation by ants may be highly underestimated. Usually they do not bite plasticine. And and another question, and these insects may also overcome the small mesh size in pollination experiments. There is a need to create barriers at plant stem level.
Let me answer first of all the first question. So ants attack plasticine prey. That has been found, especially in tropical places when they use the artificial caterpillar. But I also observed personally bite marks in Northern Europe, in Denmark. They are also easily identifiable because they are different from the ground beetles or cheliceraal mark of a large ground active insect. Of course, it's basically impossible to translate the information of ant predation to biodiversity, to density, because ants, they are colonial insects. One of them attacks the prey, calls the other, and they all come and start to tear it apart. So they are social insects, and the unit somehow is the colony, rather the individual. So but if you're not interested in investigating the relationship between biodiversity ecosystem services, if you just want to know if there is ant pest control in that system, that's enough. This is usually done in agroforestry in the tropics, and there you find a lot of ants mark. You also find other funny things like lizard, bats. I read also about monkey bites. So there are a number of papers showing that, and also there is also a publication from Australia with a catalog of the bites that can be, if you are interested in the method, you can also look at that, or I can send the paper if on request. And then they have is a photographic guide published in Entomologia Experimentalista Applicata, and you see different predators. Paolo, can you repeat the other question? I forgot.
The other question is the problem that ants can overcome the small mesh size in pollination, growing through the stem, and there should be a barrier in the stem to avoid ants to go up through the stem.
Personally, I haven't observed that, but I will be surprised if the ant goes on the strawberry flower. Just normally they go around. Sometimes they also provide pollination, but they're mainly doing other services like seed dispersal, decomposition. Yes, it's true that the small ant may enter the nest for and climbing on the pot and going to the flower, but it looks a little bit artificial. Why they should do that? I think pollen and may be more interesting for other groups, other Hymenoptera groups. That's my guess.
Okay. You have to.
Also, sorry, Paolo. Can I add another thing? Also, the possibility that ants enter the two setups. It's true that perhaps a small ant can enter, but this is the same for the open one. So somehow there is the background comparison is there. So if ant wants to go on the strawberry plant, they can go in the one that's accessible. Likewise, the one that is closed. So there is no problem with that. As far as the intention is to compare the two habitats and not the two setups.
Well, for the people that is not experienced with this kind of approach, I must to say that this is not this is a experimental comparative approach, but it's quite time consuming. It could take months to set up all these things that Marco showed, that growing the plants for pollination, growing the plants for herbivory, preparing the plasticine, preparing the seeds, everything, going to the field, repeating this process day by day in different replicates. This is really a time-consuming process, but the results are, of course, not perfect as we were able to see what is going on in the nature, but it's impossible to have a camera filming all the things that is happening in nature. Of course, this is a simplified approach, but it's a comparative approach, but still is a complex logistical experiment. And of course,
Also, if I may add one thing, Paolo, is that we are trying to measure multiple assessments. This is rarely done. People use this method individually. Instead, we are trying to get all this information simultaneously from this habitat. But normally, and probably because it's complicated and takes time, and also because it's enough to have one of them to have a publication, people will focus on pollination or on seed dispersal or on predation. Here we are trying to do something big somehow, trying to have a multiple ecosystem services assessment. Sorry.
There is a final question from Noeline. She asks if this kind of if you have any evidence of other studies that compare this kind of artificial caterpillars and this kind of approaches with real measurements in the field of this kind of services with nature animals.
There are with both in the field and in the lab comparison between sentinel prey that are real versus sentinel prey that are not real, and that they can provide different results. So the point is not, even when you use real prey, you're not measuring real predation. You're not having real density. You're not creating the real conditions. So the idea, whatever method you use, is to have a comparative result and something that you can interpret. So you want to measure predation in two habitats and compare between them. Then the important thing is that if you want to use real prey in vineyards, then you have to use also real prey in the orchards. None of them will tell you exactly what's the real predation rate in the field, because even if you use a real prey, the prey cannot escape, cannot move. I mean, caterpillars normally don't stay 24 hours in the same point without escaping. So even if you do it with the real prey artificially, it's still artificial. Of course, you may attract some predators that you may not attack with the artificial prey. For instance, you can get parasitism, right? Because you can get parasitoids, and parasitoids are possibly not interested in artificial caterpillar. You can get spiders that normally attack only prey that are moving. So you can get different results because the range, the share of the predatory community that is interesting in the type of prey you're selecting is different. So if you use eggs instead of caterpillars, you will get all the eggs predator. If you use a caterpillar, you will get the caterpillar predator, of course. So you have to think a little bit if you want to measure something in particular. So you have a species model, or such as in this case, if you only want to have comparable results, and in that case, you can pick one of them, the one that possibly is easier to standardize. In our case, the plasticine caterpillar. We like that because we don't need to have a colony for that, because they are not real. We can just buy the plasticine. And also, we like very much the fact that we can identify the predators, because when the prey disappear and then you don't know if it was a bird, a rat, or an insect. I mean, how can you do habitat manipulation to support natural enemies if you don't know who are some idea about at least which field are the natural enemies? I hope that answers the question.
Thank you, Marco. I think we are having the limit of time. Thank you for your presentation and to be available to respond to all these interesting and nice questions. Thank you for everyone by assist. And if you want to know more about this agro-eco-services project we are having here in the Azores, you can contact Marco and myself to clarify, and it will be fantastic if someone wants to replicate this kind of work in other parts.
Absolutely. We can provide the protocol and assistance for that.
Okay. Thank you for everyone to be here.
Thank you. Thank you. Thank you again for inviting me. Bye.